It is clear. The AirPods is also connected. Okay.
Good for you. Oh, okay. Just second year BSM student. Yeah. I was wondering, what about you? I was wondering, I sent those mails because I can't recognize many of the students first. Yes, I think somebody has joined. Who is that? So what I was thinking is, I wanted my students to come also. I called them. Yeah. So yesterday you talked about FIS.
Yes.
Which is a bridging protein.
Yeah, FIS, HMS, FIS are bridging.
FIS and HMS. Okay. So the point is, and they can join sit anywhere on the DNA. So I have two questions.
No, FIS actually has a sequence preference. FIS actually has some specific sequence that they prefer to bind.
And how common is the sequence?
Let me see.
My question is actually
how many sites?
No, not exactly. No, not how many sites. I mean, along the chain, how many points can FIS attach? Because it's finally a bridging protein, right? So there are two questions. They're all related. FIS and
FIS expand. Oh, I think they're much higher. FIS, the binding motif, it's about 1,500 across the chromosome.
They're 1,500.
Yeah,
so
and they are fairly yeah, they are. In the high C data, it is showing that if you delete FIS, you see the reduction of those small scale interactions. I think that scale is more like in the, oh, they actually did not do FIS. Oh no, they did. They do 100 to 100 kb interaction. Deletion FIS decrease 100 to 400 kbps interaction, but then you have those smaller interaction actually increase.
So, okay. So 100 kb, what does it do? I mean, does it form loops of 100 kb? No, you didn't.
No. I mean, we don't know. We don't know. This is just based on the high C data. If you delete FIS, you saw the reduction of interaction between 100 to 400 kb.
100 and 400?
Yeah, between 100 to 400. But then you saw increased short range reaction. But
most probably I don't understand when you, so are you saying that there, increased interaction of 100 to 400 kb is lost? But you say
no, just relatively they're more abundant. So if you lose FIS or any one of those that you NAPS binding proteins.
Oh.
While you, except for HU, you lose interaction at this, you can call the media scale, 100 to 400 kb interaction. But they think the enhancement of the small interactions is because you simply lose all the others, then you show up more of those smaller scale interactions. So it does not define the loop size, because I think many of those, for example, like the phase type of loop, I think they could be even smaller than that.
It can be smaller.
It can be even smaller because they cause some DNA looping. That could be in the range of few kb, the actual loop. So I don't know. Yeah, I don't know if that actually show up in this type of high C interactions.
So do you find you always say that this is, so you don't know what is the size of the loops, but it will be order of 5, 10.
If you just simply take the phase number of sites 1500, right? So 1500 divided by 4 meaning, basically you get another about 4,000 loops.
Okay.
So it would be a few kb in size if everyone make one.
Only if
yeah.
One. Okay, so this can make smaller loops.
But most of them, they bind into promoter regions. Phase is has a very large role in transcription. It's actually facilitates the ribosomal RNA transcription.
I see. And whenever that is happening, then another chain might come and form a bridge.
It could, but
only if they are close together, they will form a bridge. If there is suppose there is phase, and this guy decides to move and hide, and by chance touch, then it will form a bridge. And if it is not, it won't form. Is it like that?
That's what I think it could be. Yeah.
Okay, so that's one thing. The other thing I had.
But you know, one thing that people have not done any pull down experiments. This pull down experiment called the high chip. So it's a combination of a chip with high C. So you pull down on the
chip measures how much transcription activity is there.
No. Chip is chromatin immunoprecipitation. So you use an antibody, you pull down a protein, and that protein binds to DNA. It will bring down the piece of DNA, and then you can sequence this piece of DNA to get its binding sites.
Yeah. Hi, where are you guys? Hello. Yeah. We are about to start. Yeah. Oh, okay. Then just wait. So chip.
Called the high chip. It's a combination of a high C and chip. Chip is you pull down on the protein, you get the DNA binding sequence. So that's how people identify the DNA binding sites on the chromosome.
Okay, DNA. Okay, you are identifying DNA binding sites
using the protein as a bait. You use an antibody, pull down the protein from your cell. Antibody recognizes the protein specifically. Then the protein binds to DNA. Then you bring down that piece of DNA.
Bring down means
bring down like you isolate those DNA fragments. Then you can sequence those DNA fragments. Right, to get their sequence. But the combination is that you pull down all those protein, then you have DNA fragments around, right? Then you ligate them, then you do the high-C experiment. Then you identify protein-mediated spatial interaction. No one has done that. I'm trying to convince my students to do it, and no one has wanted to pick it up.
Okay. But
so then that would answer this question.
But people have done it. Your group hasn't done it.
No one has done that in bacteria.
Okay.
No, people have done that in eukaryotes, in mammalian cells, but not in bacteria. So we have been hoping to do that experiment, but I couldn't convince. Okay.
Maybe you can.
Can you guys hear me?
I can hear you.
Okay, good. So shall we start?
Yes, we can start. But before starting, I'll just ask a question.
Yeah.
So what I've been thinking is
Okay.
there is this movement
Mhm.
which is extruding. Let's draw.
Okay.
So suppose so this is a DNA,
Mhm.
and here there is one movement side. This is one movement side, and it is extruding. It's pulling.
Yeah. Well, suppose they are in dimer.
So they're always like this.
Yeah. So yeah, or you have two dimer right here. They're extruding, pulling from both ends.
So they're
That's what I suppose. Yeah, that's what people are thinking.
And if now there is a plectroneme here, there are 5 plectonemes. So you said, I mean, if I understood right, this size was around 1.10 to the power 5 base pairs.
So 100 kp, 100 kbp.
Yeah, so that's 100 kbp.
Well, based on their calculation, they think it's about 30 to 50 kbp, you know, in terms of those loops, but probably the same order of magnitude.
So, but if there can be some plectonemes also,
Yes.
then it will go up. I mean, that's the question. So my broader question is, just by extrusion, it is changing the loop size, it's changing the loop dynamics. Is it changing it only at 50 kbp, or is it possible if there are plectonemes sitting here, it can affect larger amount of reorganization? That's my question.
What do you mean when you are pulling the DNA, whether you deform those plectonemes?
No,
or
you're not deforming. No. See, these plectonemes would also have to move, right? So suppose I have a plectoneme here.
Right.
So because it's extruding, this can be rearranged. So essentially your DNA organization,
but not necessarily, right?
Changing much more than standard diffusive dynamics. That
yeah, that's for sure. I'm pretty sure those are going to form plectonemes.
Because
I think so. Yeah.
Have long-term effects because you're changing. So that's a point which I'll really like to discuss in the evening after this. But it was wondering how much these local actions are changing polymer dynamics. So like as Mipin was saying yesterday that this is diffusive dynamics. I want to say no, because of this there should be a lot of active dynamics.
Yeah. I agree. I don't think it's going to be completely diffusive.
Yeah. So let's start. So we'll
all right.
Talk, more along this line.
All right. So Nathan, did you have a question? I heard something.
No. I was just making a comment, but it's not important. Yeah.
Okay. All right. So. Any question from yesterday? All right, I'm just going to finish discussing the chromosome organization in the other two organisms. So we talked about E. coli yesterday. So today I'm going to talk about the two other modern model organism. One is Bacillus. So Bacillus, you probably know, it's a gram-positive or monoderm bacteria, very, very thick cell wall. People like this species, this organism, because it's a model organism for gram-positive, but also this one form a spore. So when it's starved, this cell just simply the form a spore inside the cell, and that spore can sustain a lot of stress, and then you can revive. So people use this one as a developmental state to study this type of regulation. But for our purpose, here I'm just going to discuss about the chromosome organization. So if I draw the high-C map from what people have done before, so this is, about, so if the middle is ori, side is ter, because this is again circular DNA. What you see, actually, you see a principal axis like this. So there are lots of little squares there. It's very, very similar to E. coli. So those are those people call the chromatin interaction domains. But besides this one diagonal, you actually have another one. It's less intensive, but obviously over there. So now you can think about why you would have another one, right? because those two arms together, you can think about this is circular DNA, right? So if you walk along the chromosome, you have lots of interaction. That's this one principal line. But now if those two arms are actually zipped up together like this, you have lots of interaction between. Now you see this part of your terminal domain would interact with your original part, right? So that's a very typical and very distinctive feature of Bacillus compared to E. coli.
That's there for C. crescentus also.
The what?
The other diagonal is there for C. crescentus also.
Yes, for Caulobacter. Yes, I'm going to talk about that. Okay. But what's interesting about this one, I think, is that in addition to those two, they found another one like this. High interaction. So this is Ori, right over here. So this indicating there is some another large scale interaction in their chromosomes, which is actually quite amazing at that time when people saw. But typically when you saw those two, you saw, okay, this is not surprised, but why you have something there, and only here, not here. It's a little hard to visualize spatially, but this turn out they show that this is because in the Ori region, if the chromosome is like this, if this is Ori, okay, they have another site called par S. And this turned out to be very important. This is exactly site where the par S is. And when you have this site, they think the chromosome form a branch over here. So then you have actually interaction within this one. This is like a long single one arm, but they have interaction over here. And this one, why it's not straight but curved? Can you think of why? If it's straight, means that they're really, 1-to-1 ratio lined up more or less. But if it's not straight, probably means, they're like a little curved. Like you have a shorter distance here, longer distance here, but then they match up with each other in this chromosome model. Okay, so turn out Bacillus has this one very particular interaction that's pertinent to this par S system, which I will talk about why this is important in the chromosome segregation. And in order to show that this is indeed due to the presence of this par S system, they actually can change this par S and move it here, there. Now you see this band start moving too. And that's a very strong evidence showing that this type of interaction is caused by par S. But why it is caused by par S? This again turned out to be related to your favorite protein, MACBEF. And the idea is that the par S sequence and also the par B, there is another protein called par B. I will talk about this a little later. Help the loading of MACBEF, somehow recruit the MACBEF. MACBEF itself doesn't have any specific DNA binding of any, but par B can help it load, and it loads over here, so it's zip it up and loads near the origin and helping it to zip up. If you delete MACBEF or delete par B, you don't get this one or you don't get this one. So it's really dependent on par B and MACB interaction. Par B, MACB, you need to have, in Bacillus, it's not called MACBEF, they're just simply called SMCs. And if you don't have an ATP active SMC, you don't see this type of structure. So, yes. Yeah, so the loadings actually only happen near Ori and in Bacillus because the par S and par B is over. So MACB is really only loaded there. And if you change the par S, they actually change where you load the MACBEF. I will show actually this is also same in Caulobacter. In Caulobacter.
I have a question.
There is a question, okay, from the web. Yes.
So par B by itself also spreads on the DNA, and par B is also known to form these cross links with itself, and that can also bring this interaction about without even MACB right?
Yes. So the question is that part B itself can also spread onto the DNA. Part B also can bring DNA sequence together to cause this type of interaction. That's absolutely true. They did the experiment. They did it part S. They did it part B. They also did it MACBEF. So when you delete any of those, you see the same effect. Especially when you only delete the SMC, you also see the disappearance. So that means that this zipping up is really requiring the SMC.
Okay.
That,
okay.
Yeah. So they did all. That's actually a very, very good question. And although I have to say that the part B, when they spread on the DNA, when they start bringing different segments together, that usually is a short distance effect. Usually it's in the range of about 10 kb. Only when you overexpress part B, then you get to really folding everything together, become this type of condensate-like structure. So on their times length scale, this is really dependent on the MACD, the SMC structure, the presence.
Okay, thanks.
Equally, MUC-PF could be loaded anywhere, but here,
no,
at specific points.
So the question from Appertine that in E. coli is MUC-PF loaded everywhere? I mean, I guess it is loaded everywhere. So the model is that there isn't any part S, part B to load them. So they could load everywhere, but people think they're loaded near the origin, not because you have an active recruiter, but you have an active regulator, which is the MAP-P. So supposedly that MUC-PF is loaded everywhere, but then just simply gets excluded from the TER region. Therefore, their distribution is towards the ORI.
But like here, you need part B to
load. Yes,
Can
you do.
Also load at other places for B subtilis. Or is that really for B subtilis, it will load it only at that point? Okay, that I do not know. But if you delete part B, you don't see this zip up. So I assume you don't see even this interactions at all. So that's where I think in Colobacter and also in Bacillus, people think part B is really the one that loading those SMC. Because if you delete part B, you don't see any of those. Why is this difference between why would MUC-PF behave differently in B subtilis? I don't know. I don't know. E. coli again, it's very odd. So I would like to have this. I think I like the hypothesis that MUC-PF itself has a DNA binding affinity, even though it's non-specific. It can load down to DNA just by itself, but once part B, you should enhance this process, in a much more efficient way. I think that's probably the most likely possibility. But apparently, in Bacillus and Colobacter, if you don't have part B, this process is very, very inefficient. Number of MUC-PF in the pre-subtilis. In E. coli, it was around 200 to 400. Here is it. I don't remember. I don't recall the copy, but I do not believe that there will be a very high copy number. It will be the same range. No, probably in the. Maybe no. I don't know. I assume that probably will be in similar range. But I don't think going to be like 10,000. I also don't think going to be like 2, 5, or 10. I think probably going to be in a similar range. Yeah, they're very, very conserved. They're very conserved protein. Like even going to eukaryotes, they're very the structure-wise, they're very similar. Yeah, they're called different. I think there are some slight accessory protein that's maybe different, but it's very similar. But in Colobacter, the structure is also the same. You get one principal line and one another diagonal line. So basically, also showing that they are zipped up. You can actually look into all those details and they figure out this chromosome is not just linear like this, but they are like a helical structure. Just by modeling the polymer and also generating this hypothetical high C delta, they think it's more like a helix structure. But what's also interesting about Colobacter, Colobacter also has this par S system.
So ori, the par S is also right nearby. And they did something very interesting. When they move this par S to another position, this or the chromosome, that they actually can move this line to here or here. So which means that this is where you start zipping up the chromosome, not the ori. The only reason you zip up this way is because pars is very close to ori. But if you move pars to other places, your chromosome starts zipping up right near the pars. That's another strong evidence showing that's the place for them to load those MSC protein to zip up the chromosome. So those are just the very brief overview of those two. And another thing that Caulobacter is also the one that really showed that transcription is the major organizer for the chromosome, because based on their map, and also they can move transcription units around, the structure they have shown is really like you have all those practony, but only in the middle, like they're all relaxed. And this corresponding to the CID boundary. I think I mentioned that a little yesterday, right? So like in a way you can think about this like the chromatin of eukaryotes. When you open up, you start doing all the transcription. And when you bundle up, it's the heterochromatin, which you don't have transcription. And this really mark the boundary. And this was the most clearly shown in Caulobacter, and also to some extent in Bacillus, but not in the other, not in E. coli.
The formation of endospore, how they act in the chromosome condensation?
Okay, so that's actually a very different question. So during the sporulation, the chromosome orientation actually is quite different from this vegetative state. And they also have a DNA translocase sitting at the inner mother pore, start reading in the DNA from mother cell to the daughter cell. And the packing of the DNA in the spore is highly condensed, and they have special protein for it, for that compaction. I don't know if their MACBF still has a big role. I think they use actually a very different system for the spore DNA packing because it's much smaller. Yeah. So based on this type of high-C experiment, so the current model of all those three organisms, they are like this. Like E. coli, it's really more or less, like oval, and Bacillus, it's like this helical structure, and the Caulobacter is also this helical structure, but it's more ranced up. So you have two arms zipped up. E. coli doesn't, but along those you have all those ammonellid planktonins. So this is what people call this bottle brush model. So you have a bottle brush, then you have all of those plankton aminoacid from the chromosome fiber. So that's how people think of what the chromosome is organized these days. So for those three typical model organisms. And there are other bacterial species people start working on more. Some of those bacterial species, they have multiple chromosomes. They also even have linear chromosomes. And it's actually very interesting, this type of high-C method. You can really map out all the interaction within the chromosome and outside the chromosome, inter-chromosome interactions. And many of those also depend on the par-S, par-B system to organize them together.
I have a quick question.
Okay, there's a question online. Yes.
So this difference in the organization in E. coli, Bacillus, and Crescentus, is it due to par-B? Because E. coli it doesn't have a par-B-S kind of system that is well established, right? Yeah. So is the zipping because of par-B then solely in Bacillus and Crescentus?
Okay, so the question is that the difference in E. coli's chromosome organization versus the other two, is that because par-S, par-B? I think so. I think so, because E. coli is really the odd one. It doesn't have this par-S, par-B system, which I'm going to talk about in the chromosome segregation system. And also appears that, the only reason that in Caulobacter and the Bacillus, they're zipped up because your MACBF load over there, then you zip up 2 chromosomes together. But for E. coli, you zip up 1 and just extruding loop along 1 chromosome. You're not taking the other one. I don't know why that is the case. I suppose that you must have some accessory protein to help in this type of configuration. But that is a major difference. Any other
Okay.
questions? Okay. All right. Any other questions? All right. So then we should go to chromosome segregation. So now we know how those chromosomes are more or less organized. And the question is that how do you separate your chromosome after replication? So when that I think we should first go over the arrangement of the chromosome. This actually turned out to be something quite interesting. In different species, you have a different arrangement. For example, in E. coli, where you have, if I draw the cells like this, if I draw my chromosome like this, then let me do. Then my ori will be in the middle, and my ter will be right here. So many of you know this is a slow growth condition. And when the cell elongate and start replicating, you usually end up ter is still over here, but you have the 2 replicating chromosomes. You have like 2 ori. And you have the ter still in the middle, more or less. Okay. When you have fast growth, you end up with more chromosomes like this. But here, the probably because the multiple chromosome organization, they're more towards the pole, and you still have ori in the middle for E. coli. So you can do this just by putting in some markers and just see along the cell cycle where those sites are. But looks like they're more or less, in the middle or quarter cell position there. But for bacillus.
But as per Carl, ter seems to be localized initially and only after 20-25 minutes.
I'm sorry, say again.
So as per Paul Biggins' data and Subjun, ter seems to be more localized the first part, and then later it comes in the middle.
Yeah, that's probably the
Fish data.
the unzipping, because I draw ter just as 1 dot, but the ter is actually a region. I probably should do like this. And only during the replication, it will unzip, it will open up. Then it will come back again because you have all those map sheet. Yeah, you have to open up, right? So many people draw ter just as like a 1 dash line.
Only at the end when that part is being replicated, but during the rest of it, ori remains in the middle for around 20-25 minutes.
Yeah, then split. Yes, then split over there. Yes. Well, I don't know how you define the ter. Depends on how sharply you want to image it, right? If you're just, for example, just putting it to one side, you can get a very sharp fluorescent spot. But if you look at this, the ter region is about 800 kilobases. It will spread out during the replication, especially when your mother chromosome being reeled into the replication fork. So it really depends on how you image it and what you define as a ter, as a domain or as a single site.
Oh, okay.
Right.
But I think when they put a fluorescent molecule, fish, how big a region does it cover? I mean, in terms of contour, in terms of base pairs.
It depends. Usually people do like about 1 kb in length. So in that sense, it's very, very small. So it's very, very small. Yeah, it's basically like a DNA site. Yeah, unless you do multiple regions, like, a few tens of kb apart, then that will be cover larger region. Yeah.
But typically they can put, these Subjun and Cast, I think Paul Biggins put at least in 2016 he had 7 or 8 points. I think 4, 1, 4, 3, 9, 8, 9 points.
Mhm.
And so you put 14 points.
In with the same color or
No,
different?
colors.
Yeah, just at the different places.
Yes,
Yeah,
different
uh-huh.
places. So you are saying, I mean, then you are really focusing where is this a particular portion, how much is it moving, right?
Yeah, you can definitely track the movement. If I remember correctly, turn region, the movement actually is quite small, not very large. Right? Is that right? I mean, turn is very, I mean, based on high C and also based on all those microscopy data, the turn region is fairly a condensed region. It will have to open up during the replication for sure, but most of the time it's just one blob sitting at the end. You know, most of the time they are at the middle. Is that right?
No, because in fast growth, as per fish data, is in the initial part of replication, start of replication, it's towards the end, and then it quickly comes in the middle.
Yeah, I know there are quite some different measurements, but I think most of the time turkey would regard it in the middle. I think during the replication, you could have a rearrangement, and also during time-lapse imaging, you do see some cells have turned, like being pushed to the side and coming back again. But I just don't know how typical.
Because the targets, I mean.
Yeah. Well, I will come to that at the resolution of the turn region. Yeah, turn can be pushed around, but I saw some studies showing that turns at one end or at the other end, but some other study at a different growth condition, and you just show up as different positions. I don't know how important that is. So that's the only thing, because you do see cells do different things. I don't know if that's a common thing as a turn being pushed that way. Because there is no active mechanism to do the turn, except for one single mechanism, which I will talk about in the cell division side. That turn will be tethered to the cell division protein.
Cell division.
Yeah.
Because turn is always in the middle, and then the cell division happens. And
Yeah.
Just before the cell division, the turn.
Segregate. Yeah.
And it's found there and slowly.
Okay, yeah, in that sense, I think it's coming from the side to the middle. Yeah.
And what I thought was turn remains in the middle only 20-25 minutes into the replication.
I thought turn is almost.
Not decided,
I think right after replication, turn quickly goes to the middle, because the segregation is actually recover the turn region very quickly. I very rarely see like one turn region is at the end of the cell, then goes to the middle. Yeah. No, it will go to the middle. Of course, most of the time it will stay at the middle. Yeah, most of the time it actually stays at the middle. It will go, when you right cut, it will move from the end of the pole to the middle. But I thought that time is very, very short in most of the.
Fast growth is very short. It's very short.
Yeah.
But for slow growth, ball beginnings, after application begins, ball beginnings data shows that it takes around 20-25 minutes before it gets localized.
Okay.
Before that, it's very scattered.
Okay. All right. Okay. Then.
I don't know what the other.
Yeah. Then the bacillus. Bacillus is a little different, but it's also more or less have this in this vegetative growth. It's also very much like E. coli, and I think there's some also dispute. The ori is there, and also the turn region is there. And of course, when I draw this, is just really like, when you take a snapshot of your cells, and most of the cell, what the position they are in. I mean, but every cell, of course, is undergoing different replication stages. So you could see them. Some cell shows up with multiple ori or turn region. And for bacillus, also exactly the same, almost like the same. So you have the turn region here, and when you have replication, then you have the ori on the two sides. But when you go for this type of anchored, so again, here they don't have any anchor. They have the Part BS system, but they're not anchored. But only in this spore region, you talk about when you start to doing the spore. So when you do spore, you basically have another membrane, and the DNA is just get reeled into the spore. There, people think that there are different type of orientation, and now you have some protein over here to anchor the mother chromosome. And I think the orientation is like this. So we still don't know why, it goes at this orientation when spores started for bacillus, but it appears that you do actually have some active protein to make sure mother is tethered and daughter cells chromosome is always going in the TER region first. So now there are some specific proteins here. If you want to know, there are some specific protein. It's also called like a RAC-A, but also you have the Part S and also Part B system only under this stage. Now, Caulobacter is probably the most well-studied one. So Caulobacter, it's like a crescent shape. It has a stalk over here, so it just stays over there. And their chromosome, like I said, is a helical structure. And they really is anchored, so the ori is right here. And they have a protein called POP-Z, which, you can consider it as cytoskeleton protein. So TER is on the other side. And so this is really completely anchored. So ori is always towards the stalk side and TER is towards the other. So now when this type of orientation, you would start thinking about how do you replicate this orientation after you finish the replication, right? So that's where the chromosome segregation. Oh, there's actually another one I found out. It's actually very interesting. So there is another bacterial species called this Onista. So this is a type of worm that's just recently been discovered. It's actually grow longitudinally. It's grow on the surface of a worm. It's in Mediterranean. This bacteria actually divides longitudinally because the only reason that if we divide this along the short axis, the daughter cell is going to fly away into the water, will never be able to attach to the surface of the worm. So this cell divides longitudinally, and the chromosome inside, they also takes this form with a very specific localization, which looks like this. So this is your ori facing outside and your TER is facing inside. When you start the division and the chromosome, they actually start moving differently. So your one side of the chromosome, the ori move to the other side, TER move to this side, then so when you have a little like a rotation of the chromosome, then they would finally divide in the middle, then that come back to the original site. So in a way, it's a little like E. coli, but they all have specific arrangement, and we don't know yet in this organism what dictates the anchoring. Do they also have the Paris BS system? We don't know yet. Okay. So now here, once this type of questions, this type of overview, there are questions. One is, what determines the ori or TER localization, right? That's what we have been discussing about why they always sit there. And the second question, what's their function? Do you need them? Are those type of localizations important for their function? Okay. The third question, of course, how they segregate. while allow function. So those are the basic questions. I think, even though I think we know a lot these days, those are still active questions people are still trying to figure out, right? So now we know some of those questions. So I will tell you a little about what we know for some of those systems.
Imaging or fluorescence different methods give the same answer.
The what?
So whatever you told about Uri, enter, and organization, is it imaging? Is it fish? Is it?
Most of those are imaging, fluorescence microscopy, not by fish. Well, fish is type of imaging, but most of those studies are done by fluorescent protein fusions.
Okay.
Light cell imaging, not by fish. Yeah, if I remember correctly. Yeah. Why?
The next question is, I have looked only at fish data because I didn't know.
Mm-hmm.
So are these different methods give consistent?
They should. The imaging method should give you very consistent results, but the growth condition can give you very different results. The growth condition you use to do those imaging could give you very different results. Because if you do the exactly same growth condition, then you do this live cell fluorescent protein imaging versus fish, they should give you exactly the same result.
Why would people change growth conditions when? So what is the reason for people adding complications?
I don't know. So usually there are, for example, for E. coli, there are multiple growth conditions, sometimes just for the experimental convenience. For example, room temperature growth is much easier than 37-degree growth, right? Because you have to control the temperature, you have to incubator, make sure you have to keep the temperature consistent.
Why would people do at 37? Is it fast food?
Well, E. coli supposedly the optimal growth condition is 37 degree and at LB, which is a rich complex media. So that's like a mimic how the wild type E. coli could grow. That's the fastest growth condition, which is about 20 minutes per doubling time. Yeah, so
that's
37 degree, yeah. But LB media is a complex media. It's yellowish. It has a lot of fluorescence background if you want to do imaging. So people who do imaging try to move away from this type of media, and they use defined media. They use minimal media, or minimal media complemented with some nutrient, so that you get a clear broth. Then it's easier for you to do imaging. But then, of course, those media will not be able to provide the same level of growth rate as LB media. So then cell will grow slower.
Just repeat why 25 degrees is better?
Well, it's just easier. You can just grow the cell in your lab without any incubator, like room temperature. People call it room temperature. So you don't need any special equipment. And also you don't have any temperature change when you take your cell to your imaging station. You don't give a temperature shock, so you keep all the condition consistent. But if you do 37, you have to keep your imaging chamber to be at 37 to be heated up as well. So a lot of those are practicality for using different media. So most of the people who want to do imaging, they use minimal media or rich defined rich growth media, so that you have a clear broth, but then the condition will be different from LB. So that's why there are lots of different growth condition, and sometimes you really have to look into the fine print and see what's the condition used. Okay, all right. So when those questions in mind, so let's first talk about the most textbook type of chromosome segregation, which is in Caulobacters. Caulobacters, if you remember, I draw the cells like this. Well, this is not good, but anyway. So we have the chromosome like this, and we have the anchoring system. Let me put another one. So we have Ori here, right nearby. And what you have is that you have near this Ori, you have this part S sequence. And part S actually binds to another protein called part B. Some of you probably actually know about this one. So in a way you can think about your DNA segment over here. You have a part S, then you have part B bound down to it. And this part B protein turned out to be most important for the segregation because you have another protein called part A. It can be a little another protein called part A. So let me just label. Part A is an ATPase. And part B turns out to be a CTPase. So it's another enzyme that hydrolyzes nucleotide. When cell grows, so part A is actually bound everywhere on the DNA, fairly non-specifically. Then what happens is that this has been worked out. When cells keeps growing, you have, so let me draw another one. So you have your stock over here and part A. So when you have a new replication origin coming out, this is the older one. And this is the third region. Oops. This is the third. So when you have this new ori coming out, this ori, because it has a part S system, what happens is that the part B loads, let me see. Part B actually is a dimer. It can actually load onto the DNA at the part S and clamps on it. And this is because you have the part S system over here. Once it loads onto this one, it can slide, it's like a clamp sliding along the DNA. Now, when it encounters part A on the chromosome, which is bound to the DNA, on this little carpet of the DNA, it's going to stimulate part A's ATPase activity and causing the part A to dissociate from the DNA. So in a way that you create a depletion zone over here. So part A here are still bound with the DNA and they have ATP. But when your part B come over here, it hydrolyzes, stimulates the hydrolysis of part A of ATP, then just deplete this region. Then what it does is just continuously doing this, like part B is continuously chasing part A across the chromosome. And this chasing interaction move the ori region to the other side. So you end up when this ori gradually coming to the other side of the cell when the turn start moving back to the middle. So this type of system, part B stimulated ATP hydrolysis of part A, it turned out to be a self-patterning phenomenon. There are lots of models to go into it. It's constantly like you don't need any motor protein inside the cell, but by this part B stimulate part A dissociation from the DNA, you can actually create bias for part B attached DNA fragment, and that allows the fragment, the DNA, to continuously treating along the part A filament.
The part B is following part A.
Yes.
Why is part A going to that side?
Part A is actually not going. It's just purely part A is diffusing. It's diffusing all around. Part B comes in, creates a zone that does not have part A. Then part B start going for the next one. That's create a bias in the symmetry. So this is also plasmid actually segregation mechanism. So you can actually recapitulate this whole system in vitro. You get a plasmid, you get a part S sequence, you add part B, then you add part A onto your surface, then you're going to see the plasmid start moving. So it's a completely self-contained, it's like a motor protein based on polymer dynamics. Yes. No, PAR is supposedly to be just like a blanket of polymer over here. And when there are actually lots of theoretical modeling go into it, if you are interested, what people think what happens, this is like a homogeneous PAR, but when PAR B comes in over here at this position, it's deplete PAR A. So this part of PAR compared to this region, you have less, you have more, and that is a symmetry breaking. That is continuously tagged on the next region where you have more PAR A. And it yes, yeah. And they think there is a slight bias. So you can go, sometimes you can see the plastic going back, but most of the time the plastic is going zigzagging but towards the one direction. But you do have some thermal fluctuations for that. Yes. What happens to? Oh, then it doesn't move. So the question is that if you remove PAR A, does this work? No. If you remove PAR A, PAR B is just going to stay there. You have a what?
You not have PAR A close to your old because it's a PAR S.
Yeah. You would. It's just this recycling is not very fast. You actually create a depletion zone. So when PAR B comes here, it actually creates a depletion zone. So inside the cell, that turns out to be actually fairly large. So you end up, that's what people think, you end up with this half gradually depleting of PAR A, and only this part having PAR A. So this one continuously chasing, PAR B continuously chasing PAR A until to reach the other side. It's the same as in the plasmid segregation system. And I mean, there are lots of other things going on. For example, PAR B is a CTP. And when it's a hydrolyzed CTP, this clamp opens, then it fall out from the DNA. So there are lots of also dynamics coming out. And you can actually put in all those numbers. There are quite some good measurement, biochemical number measurement to show how this system actually could work just in vitro, purely based on those diffusion reaction constant. So this is the type of chromosome segregation that's very specific to Chlorobacter, because Chlorobacter has this segregation system. Now there is another type of segregation system, which is actually similar to this, but it's slightly different. It's more close to eukaryotes. So that is not pertinent to chromosome segregation, but it's used in plasmid segregation, which I think actually are very similar. So I'm just going to tell you that one right now. This one, I think we actually have experts here. This is the RM system. So if I draw, so this is a plasmid. So this is for this type called the R1 plasmid, and we do have expert here to understand it. So what they have a sequence here called PAR-C, and this sequence binds a protein, again very similar, called PAR-B. So those are all acting homologs. But this one is interesting because it doesn't like chasten PAR-A like we showed last time, just constant hydrolyzer. It actually binds to a long filament that's made by another protein called PAR-M. And this grows, so PAR-M actually start growing when you have PAR-B over here, just continuously grow. And until it reach another plasmid, have the same, it has the same PAR-C system, and also have PAR-B. And this is such an interesting system. You can actually do this in the test tube. You can have a DNA plasmid, have those sequences, just add those protein. Now you are going to see that this protein, if you label it, it's going to start growing, then it's going to contact another plasmid over there. Then after it's contacting this plasmid, then what's happening is that this filament continuously grow and just simply propelling the two plasmids away from each other. So this is another segregation system, just so elegant. You think about, again, 3 system, and the polymer actually, the monomer parm is actually added at the end, because you can photo bleach the middle part, they do not recover. So they just simply add monomers at the end. And this adding is more like a Brownian ratchet model, just keeps adding on, then that's how you separate everything together.
This plasmid and this plasmid are copies of each other.
Yeah, they're exactly the same.
But this plasmid could diffuse around. So how does it figure the direction?
No,
Or it
there
tries
is
out
no,
of-
yeah, this one can go. If you have multiple, this one only one plasmid, they only put in one Parse sequence. You end up with one filament. And it's just like a random fish. Like if you encounter another one, they can capture it. And at the same time, this one can also grow and capture another one. Yeah, so they actually can grow into,
randomly.
if you do, this is an in vitro, the experiment I described is an in vitro experiment. So they did everything in the test tube. And you can actually see they actually can grow into a network, because this
But
is a
if it doesn't
bundle.
touch another, does it break?
Yes, it will break. Yes, that's a very good question. So this one, if it doesn't reach another one, this one at one point it's going to collapse. It's the people call this dynamic instability. They just simply collapse and coming back. And if you cut in the middle, the filament will very rapidly shrink, depolarize. But if you have another site over here that stabilizes the enzyme, they can stay for a long time.
So only once they touch each other, they will grow and push.
It is amazing. If you guys are interested in this, I would really suggest you go back to the original paper. They did this in a test tube, and you can even use its filaments are so long. You can actually see the filament to grow into a U shape based on the shape of your tube and propel this one. So you just put this system in a test tube, and the filament is so long, and they can grow based on the U shape of your tube and pushing the DNA. Pushing the plasmid on the side.
What is this, like, this growing out from this finds the other, like, there has to be a orientation. It has to be rightly oriented.
It has to be what?
Rightly oriented in the,
What do you mean oriented? Like,
if
It tries out randomly. That's what I asked. And then I have so it tries.
it goes all different places, and if you encounter one, the capture and stabilize. So we can think about this constantly going out, shrink, going out, shrink, going out, shrink until you attach to something.
What is the size of a plasmid? The reason I'm asking is
okay.
because it will also have angular diffusion. So if it has the angular diffusion, will this arm do like this?
Oh, that I do not
So
know.
what is the size of a plasmid? But we can calculate what will be the approximate angular diffusion.
That I do not. I assume this is a regular plasmid, maybe a few kb.
A few thousand kb?
Yeah. Oh no, a
Let's
few
say
thousand base
10,000
pair.
base pair.
Probably more. Usually the typical size of plasmid 2 to 4 kb.
But
2 to 4 kb.
Oh,
2 or 4,000 base pairs.
small. Oh, then there will be a lot of angular diffusion also.
I
So
don't
then
know.
tell me if there is an arm like this,
Uh-huh.
and I am a plasmid, and because I'm going to move, am I going to do this also? Because this will
I
also
think
help
you
me to
can.
search another
Yeah,
guy.
yeah. No, I think you can. I think in the movie they put everything down on a slice. I think they use some sorts of a coding so that most of your filaments will be like in parallel on the slide. But you do see sometimes the arm going up because the focal depth you can see a little. But most of the image is along the surface. You certainly can go up, but if you go up, you just can't really image them. Yeah.
You can't image them, but that doesn't prevent it.
No, it doesn't. You
because
do
that
see
will
some
help-
of those.
because that will also help find the other guy.
Oh, you mean actually you can actually fish something from the solution and bring it down?
I am a plasmid. I have to search another guy.
Uh-huh.
One is I just do this and come back, do this and come back and search.
Yeah.
The other thing is I do this, and since I have some lifetime and I'm doing some angular diffusion, I'm also
Yeah, you
searching
could.
like this.
Yeah, I think you
That will
could.
speed up the search process.
Yeah. Yeah, inside the cell is
But
much
I
less.
think
This
that
is an in vitro.
the distance between the two will be much smaller than a cell, right? What would be the distance
Yeah,
between?
yeah, no, but I'm saying the polymerase direction of particles is
What are the dimensions? Or what would be the average distance?
Micrometers. This one can go for micrometers. Now you can see them, I think 10s or 20 micrometer in the in vitro system. Inside the cell, of course, it's much smaller. You don't need that. But in this in vitro system, you can
But
see
since
them.
this is replicated, it need not be very far away, right? I mean,
Well, this is an in vitro experiment. We just mix all of those together, just see what happens. Then what they saw, that you can't adjust concentration, everything, of course. Then some of those start growing on the plasmid, then you can see you can capture another one. Yeah. So this is a very interesting type of plasmid segregation using actin. So those are actually actin homologs, paras, those systems. I will talk more about that skeleton tomorrow. But there's a third one. Yeah.
After this is like done, so there's a critical distance after which it will break down.
That I do not know.
So after it has found its partner or something, so will it break down?
It will eventually, because this is just a filament. So once you finish hydrolyzing all the ATP, it will break down. But I don't know how that happens. So I don't know if it's where when you have cell division or some other things, or this is just purely a random process. Yeah.
we could maybe write down the author name, because if we need it, we can at least go and search.
Yeah. I think the first author
Because
name
we
is
recorded also, right? So we'll have a reference.
Ethan Garner. This paper was done, I think it's in a science paper, I think 2007. Yeah. But if you just search par M, I'm sorry, this is not par B, this is par R, this protein called par MRC system, then you can find this one. Yeah. It's very interesting. Actually, not many people have done model on this system. The par SAB system has done lots of modeling already, so I don't recommend you do more. But this system is very interesting. No one really has looked at much of the dynamics. Modeling, like,
I mean,
well, the rate, whether the ATP conversion would really drive this motion, and does that suffice to push the plasmid aside efficiently during cell division?
so
or-
you have to put in ATP concentration in the model as well.
Yeah. You need to. Well, with this system is probably simpler than the par AB system, because that one is really complicated. You have a continuous, like a ratchet model. This one, I would assume it's also a ratchet model, because the monomer is added in between. So basically you would have a constant how this one break off from the par R system and still close enough that you can add a new polymer. We use like a microtubule, that's how you actually push things apart. So I don't know how much similarity you're going to see, whether those numbers would add up.
And okay, then there'll be different concentrations of par A, par B, and it has whatever.
Yeah, par M,
R.
par R, and also ATP concentration. Yeah, the binding, the capture efficiency.
Oh,
So those are two types called type 1, type 2, but there is another type, which is FTSZ homolog or tubulin homolog. So you can see the bacterial cytoskeletons are actually very different. This one is called type 3. It's mediated by the type Z system. So this one is, again, it's also another type of plasmid segregation system, very similar to the second one we talked about, but slightly different. So this one is also have a sequence, plasmid, then you have a sequence called type C on a plasmid. Then you have one protein called type R, very similar to what we just talked about. Now the protein that's really polymerized is called type Z. This is the tubulin homolog. It's also formed this long filament along the plasmid, but only forms one because it has a polarity. So this filament depolymerize on this end. It's treadmills in a way. And this end is polymerized. So in a way, it's very similar to FTC, which I will talk more in detail tomorrow. So now can you see how this one works? So you just constantly grow over here, then your plasmid is constantly chasing type Z. And this is how this whole thing is moving forward. And this is only found in a very rare, those Bacillus anthrax type of bacteria.
Chasing, what do you mean? Is there some attraction, electrostatic attraction?
Yeah, there are interaction, type R and type Z. Those two proteins actually interact with each other. And that interaction actually slow. So if you don't have all of those, this filament will continuously do whatever it wants to do. Polymerize on this end, depolarize on this, and this is based on the GTP activity of this filament, very similar to FTSA. But if you have this plasmid with the type R protein over here, this one will try to bind this one end of the type Z filament, and this binding reduce the depolarization rate of this filament. So as a result, if you reduce the depolarization at this end, then your polarization end, it doesn't change, right? It's just a bimolecular reaction, just continuously add on, then your filaments actually grow longer. But even though this is stabilized, this end, it still induce the GTP hydrolysis, and it's also continuously, this end going to depolarize. But when you have something depolarized, this type R latch onto it continuously. So that's more like this plasmid is chasing this filament, but you can also see that this filament is pulling the plasmid. And so that's another plasmid segregation system. Those are the three major types of the segregation systems. Very active. So this will belong to the Caulobacter regime, right? You have some anchored, they're anchored at the cell pole. You have an active segregation system to move the chromosome, then everything along the chromosome is very much defined. Okay, but then what about, do you mind that I continued? Because, or I really only have like Bacillus segregation and E. coli and entropy model, or do you think?
Or you want a T, or it depends, or what do you want? Because you are the person who speaks.
Okay, it's up to, because I think I probably only need another half hour to finish. Do you want to continue?
Then let's take it.
Okay, all right.
Because
Okay.
Everybody can have a break.
Okay, then we come back at oh,
Oh, okay. No, let's give a bit more time.
50.
50.
Okay, all right.
Question. I mean,
Thanks for talking. I understood what it meant.
First year we decided.
Okay, but why should these two guys, Chrysanthus and Subtilis, load Macbeth at only one or two places? I mean, what purpose does it serve? How does it help the DNA
I
organization?
think that's actually a very interesting question. I will touch upon that, but I think where you load it, how determine your localization.
It helps determine the localization.
Yeah. It's how determine where your origin of either with the half quarter or the cycle. Because where you load it is where the DNA will start separating.
It's okay, yeah, because of the loops.
Yes.
The loops will entropically push away
Yes,
each other.
exactly.
So that's where it will start
Yes, I think
segregating.
that's the reason.
That's something we could, okay.
Let's start.
Yeah, should we start?
So the next one is bacillus. So we just talk about Colobacter, like everything is a program. You have an anchor, you have segregation machinery, you know exactly where to go, right? So it's very directional. But now we come to Bacillus. Bacillus has the par S AB system, but it's not anchored. So how does the cell actually know how to segregate? So this turned out that when this was shown, I mean, of course, this is a hypothesis. So when Bacillus chromosome gets replicated with this ori, now of course you have the two replication forks, and you have the ori here, you have the ori here. And because you have the par S on those two replicated, like what we talk about during the chromosome confirmation capture, those are the sites to load the SMC complex. Now, what's being proposed? I'm actually people model this whole concept. What's being proposed that as soon as those sites are exposed, the SMC just comes in and starts zipping up. And this zipping up motion is the one driving the segregation.
Zipping up.
Those are the new chromosomes start zipping with the SMC.
Oh, with
that is
SMC. Oh, and then it will start
So
exploding.
you
Then
know
it
when
will
you're
then-
thinking like you're replicating, the moment you're replicating, you're being dragged away. And this dragging away is not something actively pushing away. It's just simply by zipping them up, by condensing them away. And that somehow segregates the chromosome.
Have people model this?
I actually don't think so.
Huh?
I don't think so.
Will people be interested in such a model? I
think
mean,
so.
what will it?
I think this will be very similar to E. coli.
Exactly.
So that's how people think about Colobacter. But for E. coli, E. coli is even worse, right? E. coli doesn't have the PRF system at all. So that's where people start proposing this entropic model. So entropic model, then you'll be very similar. Just saying that if you have two polymers, they do not favor to be homogeneously mixed with each other, right? Because you have less conformational state for each one. So they naturally repel each other. And that's how people think that's in this chromosome segregation, how that happens in E. coli. But to have this one to happen, there has quite some actually modeling being done in Paraver or Will as well, which is that you need to have very high polymer density as they're really high. And second of all, this appears to only occur in cylindrical shape, not in spherical shape. So it's an entropical model. So the first is the density of the polymer. Second is the shape of the cell. And if you satisfy those two, you can actually satisfy the segregation of chromosomes.
We are actually trying out entropic model even inside this sphere. So what if we do this? I mean, I can show you quantitative data. So this is not driven by biology, but suppose you have something like this and a big loop. You take 6. So here there is a symmetry break. are on the outside.
Oh, really?
I have quantitative data. Yeah. So if you have three such short, small loops, the propensity for these small loops to be on the outside is more. Now you can also have this. It's relatively less. So I have quantitative data on this, and if that is useful, I mean, I'm doing it as a polymer model. I just want to look at the property of architecture. But if it is useful to biology, that would be great.
No, I think, well, here those loops, so those are just purely polymer, right? Has nothing to do with the proteins that we talk about. So I will be interested in seeing that this by itself can reproduce. And people think that just by those two, you have the chromosome segregation when it happens. You naturally have, sister chromosome over here, the third region over here. And just by the pure mass, because these are increasing polymer density, then you can actually segregate and also simultaneously satisfy.
I didn't understand. Can you repeat?
No, this is just like a chromosome, right? This is the chromosome, it starts to replicating, then you generate one sister chromosome, then generate another, and this is the unsegregated mother.
Oh, okay.
So basically just simply have this two mass, you will be able to segregate.
Yeah.
But in reality, we know this process also has to be related to the FBF. Because when you are polymer, when you have polymer to repel each other, you at the same time you're actually making all of those loops.
Yes.
So this part, I don't think has been modeled. I would love to see how much this will be able to facilitate this whole process and also position.
Let's talk in detail after you finish.
So I can give you some little experiment data people have done, which is actually quite interesting. So you can actually take the whole chromosome of the E. coli, you take it out, you basically break out. Okay, so first experiment you have done is that you take this one, you treat the cell with some drugs to change the cell shape, either mRNA inhibitors or some, you take the cell wall out, so the cell becomes spherical. Now they say that because it's a tropical model says shape is important. Now you change the shape, does chromosomal segregate or not? It doesn't. It has a lot of chromosomal segregation defect. So I thought, well, even though that's the case, it doesn't demonstrate this because when you mess up with the cell shape, lots of things messed up and the cell division can't be held up. And so that's not a strong evidence.
Okay. I see.
Okay, right. So, but what they did is okay, so if you are not convinced that's the case, what about I still inhibit everything, the mRNA? I just put this cell, without the cell wall, into a chamber, microfluidic chamber that has this interaction. Yeah. Then they start separating. I thought that's probably the most convincing evidence that I have seen.
Who did that?
It's one of those key stackers experiment called Genome in a Box. You should look at that.
Oh yeah, he has 2 or 3 of these
these
and
So I thought we can look more into the detail of that one, but I thought that's the first one because when you run it up, it doesn't segregate and you don't change anything, just simply combine the shape and this thing starts segregating again. So I think that's really the strong evidence that the shape is a big factor. And now, of course, he didn't do the experiment where you also look at the effect of BEF, but I think those will be experiments. And I'm pretty sure that if you don't have BEF, even if you have the shape confinement, you probably will not produce the right segregation.
If you don't?
If you do not have BEF, even if you have the right shape.
Yeah.
So in a way, I think the model that I think will most be reasonable will be a combination of this tropical model as with the active for compaction by BEF.
Those will do it even better. Smaller loops will do it even better. So what Sujun showed in 2010, ring polymers, they segregate. What we are showing, you put in more loops, you get better segregation and also organization. Now within that, if you put even smaller loops as Sridhar has already done, I think it will speed up the
Yeah, I think then
segregation.
you can really monitor the speed, the rate for the segregation, and also the loci, the locus position.
Loci we are already getting, but we just add, there will be more repulsion and we'll see what will happen. We'll definitely do it.
So that's for E. coli, this off back doesn't have parity system, is not anchored compared to Bacillus. Bacillus is not anchored but has a parity system.
Okay.
Then to Colobacters, the most advanced is much more advanced system. So E. coli is really something that appears that's very sloppy. But even so, it appears that it's doing a pretty good
Good
job,
job.
For this type of segregation.
Even fast growth.
Yeah, even fast growth. Okay. Now I want to give you an overview of the segregation, but there is one last step. One last step, because at the end when you're term rating replicated, many times they're tangled, and we're talking about you have all those, because if you're not BF just streaming all the DNA, at the end all the tension going to be at your end of those two chromosomes. So there is another one last step for chromosome segregation, which is the turtle segregation, which is actually quite complicated process. Oh, for E. coli again, I think I should just say that MACBEF is each arm is not ZPR. We still don't understand why that is the case. Why you couldn't just do what's actually makes this different from Bacillus. Now the last step is the resolution. So if we draw the cell onto divide like this, now you have two copies of chromosome. And they could be tangled all over the place. Like this. So how do you actually, so there are two different things. One is that you could have two DNA catenated with each other, so they're just simply two copies. Or you could have a dimer chromosome. Like during the replication, you could have some recombination happen. So you end up with one gigantic circle, but two copy chromosomes. So they're like covalently attached. So both of those scenarios have to be resolved for the cell to divide. And that's actually is based on a type of proteins. So one type of protein, many of you probably know, we call it FTSK. It's a cell division protein. It's a hexamer. It's basically, binds onto DNA.
Can you write it properly? TEP, what is it?
Sorry. FT.
Oh, okay. FTS. Yeah.
FTSK.
Okay.
So this is a translocase. It has a transmembrane segment attached to the cell membrane, and this one interacts with all the cell division components that I'm not going to touch upon today. So this is translocase that binds to a particular sequence called COMS. You don't have to know all the, but it binds onto this DNA sequence, then it actually knows the directionality, and it keeps pumping the DNA, just dragging the DNA to the other side. Then you have a GSK on the other side as well. So just keep push the DNA over, and you have to on the other side as well. So that's just simply say, okay, we're going to about to divide, you have to move away. So that's the one thing. So FGSK also play a role in chromosome segregation, not only in division. Of course, it has other roles, but this is one first important protein during cell division. Now you also have another protein. FGSK doesn't have no, it's a hexamer. Yeah, there actually was a beautiful structure of that TSQ hexamer. So what happens, you have, they're like not on the same plane. They're like slightly tilted like a ladder. So they actually rotate the DNA. It's like a spiraling up the DNA. Yeah, like helix, right? It's a very beautiful. But I don't recall that TSQ use ATP or any energy for this. I could be wrong. I don't remember. Uh-huh.
I think it all being part of the AAA ATPase Act associated with diverse AAA class of protein.
No, this one is not. This one is not. We have the AAA translocase by another sequence FTSEX. No, actually, yeah, I don't know. They do interact with FTSK, but I actually do not recall that they help in the DNA segregation. I have to say this DNA segregation role of FTSK is not essential. So you can cut it off, get rid of this part, cell can still survive, but they have defect, but they can still survive. Only the transmembrane domain of FTSK is essential, and that is required for cell division. So we still don't quite understand the role of the transmembrane domain. Yeah, there are quite some good hypotheses going on. So in a way, we think that the general sense is that FTSK is the one to deliver the information from the chromosome to the division machinery. So say, okay, now I'm pumping, I'm getting ready, you should also get ready. But we don't know how that was delivered. There are actually some other communication between division and chromosome, which I will talk about tomorrow. So this is the one protein. That there is another protein called, some of you probably also know, this actually protein binds a deep sequence. When you see this deep sequence,
Oh, okay.
yeah, that's basically your term region, right? That defines your termination.
Yes.
So there
So
are
this is a protein.
no,
What?
it's a DNA sequence.
Okay.
It's a DNA sequence, but there is a protein that binds this protein called ZRCD. So it's a recombinase. So it binds over here. So this is again, this is like a redundancy. So this is a recombinase that recombine the two copy of chromosome to separate into
Oh, it
monomers.
will, when it cuts, it will complete the circle.
Yeah, because
Oh,
otherwise, I think usually it's like 10 or 20% of your chromosome at the end of replication, they become a dimer chromosome, like twice the size of your single chromosome. So now you need this recombinase.
20%.
20% of the cell will end up with a dimer chromosome. You know, a dimer chromosome, like you have one chromosome, right? Suppose just you replicate it, you're supposed to be like this. 80% time you're going to end up something like this.
Oh, okay.
But sometimes there's some recombination happening, you end up with two copies, a dimer chromosome. Like this chromosome is twice the size. Like everything has two copies, but they are covalently linked. So you have to cut it in between to separate those two. And that's what this protein, ZRCD, is doing. And FTSK actually recruits this recombinase come over to cut it so that you can separate those two. But those are probably not the most common scenario. The most common scenario is the catenation, right? So when your two strands are right next to each other. And then you need another protein called, this is our good friend, TOPO4. You guys still remember we talked a little about TOPO4 at the beginning? TOPO4 is type 2 topoisomerase, so it's basically cut a double strand DNA to release tension. And this is absolutely required at the end of the chromosome. The reason for that is that they are catenated with each other. TOPO4 has to come over and cut it open and release them. But how does it do so? There are lots of models for this, and turns out that BEF also has a role in this. First of all, that FTSK also recruits TOPO4 to the end of the chromosome. But then what happens is hard for it to draw. But if you have a DNA loop like this, if the BF is constantly storing the loop, then you're in a way you're pushing everything away.
But
with
wait,
FTSK.
MUG-BF won't be there in the third region.
That's actually the interesting part. I know. I was thinking about why that would be the case, but I suppose towards the end when you're actually TERT being replicated, maybe that's the moment, brief moment, all the MAP, everything is gone, and you're really allowing, and also FTSK recruits those MAP come over here. So this is being pushed away, then you end up with a very tangled, very tension, high tension structure. Like you just simply this one is here and this one is right here. So they're really just like influenced like this. And people believe that is the one that's active in the top of four to come in and to active in top of four to come in. Because before you can think about if they're all tangled loosely, like there is no tension until FTSK start pulling, until MACB starts streaming everything through, tighten everything, and you end up with a very tightly knotted DNA. And that's the region top of four will come in and kind of release the tension and you separate. So that's the last step of the chromosome segregation.
By the way, because we had a model, we were also looking if you put small loops. Remember, we have that R22, putting small loops. So we were saying, okay, once the polymers are segregated, here it's more of a physics problem. Once the polymers are segregated, how much would they again have a tendency to mix, at least at the ends? And there, if you have smaller loops, they tend to I think the word is overlap, right, Sriram? Yeah. So the question
but
is.
They are like homogeneously mixed with each other? Or?
No. So the question is this. Now, suppose two polymers, put them in a cylinder, and they have decided, give me a blue chalk or yellow, whatever. So this is cylinder. Now they have entropically separated, but sometimes because there is entropy, this can also sometimes do this, right? And this guy can do this. So most of it is segregated, but the question is just like this: how much is the tendency to overlap? Spatially, because of entropic reasons. There's no, as you were saying, they can get entangled and so on and so forth. But here, at the end, if you have smaller loops like this, like you remember my arc2 loop has two big loops. These two small loops locally again prevent overlap or reduce overlap is the right word.
That's actually increase the entropic force.
Yeah, and so that they have a less tendency to overlap, and we have systematics of 12 architectures. I mean, we did it and we just wanted to do a polymer physics thing, and then we figured out that they overlap less. Now you are saying this is a huge problem, and you are saying that how you prevent overlap. So we have some data on that also.
What do you mean? How to prevent?
Prevent is if you just have big loops, they can easily get into configurations like this.
Oh, like overlapping.
Whereas if you have moved for whatever smaller loops, they are all systematically entropically repelling each other, and things like this is reduced. I'm not saying prevented. It's significantly reduced. That's all that I'm saying.
And that's how I think that why as in the MACBF also.
MACBF will do the same thing, small loops.
Well, again, that is the hypothesis because people see this type of defect in MACB and don't have the church segregation. So it was proposed, and there are interactions between MACBF and topical forehand. That's how people put together this hypothesis. But I don't think anyone really monitored or showed that.
Okay. we have data, you can see how you can use it. I didn't know all of this.
Okay, so that's all for all the new growth organization segregation. So I think I'm done for that section. I planned only for 2 hours, and we went for 5.5 hours total. But then tomorrow, the day after tomorrow, I don't know. I mean, I was planning cytoskeleton, cell division, cell shape. For that work, originally I planned for some more. I also planned for example, some imaging methods, sequencing-based methods. I don't know if you guys are interested in some of those. Or
I mean, I don't even know what I'm interested in.
I would just go with what I would just mainly talk about cytoskeleton down along the way because so related to polymer dynamics, the cell division, cell shape. I would just put them all together through the last 2 days. Okay. All right. Okay.
Thank
Thank
you.
you all.
Ask them whether they have questions.
Yeah. Do you guys have any questions? No? Okay. All right. Thank you.
Okay, thanks. I will cut the connection and anyway, yeah. We
Thanks,
can
Apatim.
हाँ
Apatim,
बोलो हाँ बोलो
do you have class tomorrow?
tomorrow हाँ करेंगे. I will open this room. I mean, I've arranged to open this room and we'll have this one.
Okay.
I mean, we'll continue.
Okay.
I don't know whether you are saturated or not, but I can simplify it for you.
Yes, I will talk to you. Yeah, after this. Yeah. And
Yeah.
the recorded lecture I'll go through again, over and over. Yeah. sometime.
Yes. I think let's talk because much of these things I didn't know the names of all these proteins and all this business. Some of which we have done with as a polymer model, and I'm going to tell her in detail about my work now, and she can find it useful. हम तो polymer model कर रहे थे. I mean, because ज़्यादा biology समझ में ही नहीं आ रहा था. So now after break, I'll tell her about this spherical thing and she might figure out things which she finds it useful because I didn't know the context. मैं ऐसे ही polymer architecture का properties निकाल रहा था. तुम्हारे साथ बाद में बात करते हैं रात को.
Sure.
क्योंकि वो जो एक अजीब सा चीज बोली ना, she said at some point, I'll just tell you. एक कोई बैक्टीरिया या किसके बारे में बोली.
Yeah. And
attached to the substrate. And once this is organized in an opposite manner. तो मुझे इस organism के बारे में कुछ मालूम नहीं था. But I was trying using polymer architecture. I was trying to play around like an Ising model types. इसको head बोलो, इसको tail बोलो, and you put it in a cylinder. Will small loops try to move towards small loops, or I mean, like, so इसको अगर ऐसे बोलो, you put it in a cylinder. कुछ preference है कि नहीं? और ये मैं अपने मजे के लिए कर रहा था. अब शायद ये चीज इधर काम आएगा कि नहीं, then other people can tell us, right? Because मैं just polymer architecture को ये करके क्या properties निकल सकता है, ये मैं खेलने का plan है. Whether it is relevant biologically or not, I don't care. अब वो तो बोल रही है वो relevant भी हो सकता है.
अच्छा.
We'll talk later. Okay.
Sure.
Yeah.
Okay.
Yeah.
Thanks a lot. Yeah.
Yeah. Bye.